Antibiotic-resistant bacteria and factory-style livestocking by David Carlton, Ainsley Gordon, Reese Phillips, and Olivia Zietz. Hi, this is Olivia Zietz. Factory-style livestock farming is a relatively recently developed technique used to maximize efficiency and output. This is achieved by creating a highly controlled environment for the animals. For example, the temperature and light exposure is highly regulated. In some cases, this can be beneficial for both the animals and the farmer. Other times, the animals suffer at the expense of efficiency. For example, animals are often kept indoors and in cramped conditions. The practice that has perhaps the greatest implications on the rest of humanity is the increasing use of antibiotics by farmers. When used frequently and improperly, antibiotics can lead to resistant bacteria, which can have serious consequences for people and animals alike.
My name is Ainsley, and I'm going to discuss some of the antibiotics that are used in factory-style livestock. The first antibiotic that I am going to discuss is called ciprofloxacin. Ciprofloxacin has the chemical formula of C17H18FN303. Campylobacter, a bacterial disease, is becoming more and more resistant to this antibiotic. Campylobacter was found in 20% of chicken breast in 1999. The drug was banned in 2005 because 30% of bacteria found in chicken breast were resistant to the drug. Colistin is another antibiotic that is being used in factory-style livestock. The chemical formula for colistin is C52H98N16O13. This antibiotic was initially used for getting rid of dangerous bacteria in livestock. E. coli, a form of dangerous bacteria, has recently been found to be resistant to colistin. The resistance of colistin in E. coli is spreading through the strains of bacteria found in E. coli. There are many types of strains of bacteria in E. coli, and usually things can only be transferred to the same strain of bacteria. The resistance to colistin is spreading through different strains of bacteria, which shows just how powerful this resistance actually is. Both of these antibiotics are becoming resistant in livestock, which is leading to bacterial diseases becoming more prevalent in the livestock.
Hi, my name is Reese Phillips, and I'm going to explain to you how bacteria become resistant. Antibiotics inactivate essential bacterial protein. These mutations in the gene can cause the antibiotics that attack bacteria from being able to bond with the proteins, thus leaving the bacteria active. Genetic change can also lead to the increase in population of bacterial protein. This means that the antibiotics cannot inactivate all of the proteins. With this growth of proteins, the bacteria are left and the resistance spreads. The bacteria that are left over may become resistant to antibiotics by developing an antibiotic inactivating enzyme. Genes also transfer bacterium resistant to antibiotics, to other genes, causing them to become resistant to antibiotics. Antibiotic-resistant bacteria can also be transferred from food, from water, or from human contact or animal contact. When antibiotics attack, some bacteria are resistant and can survive the antibiotics. They can grow and thrive because more resources are available for the bacteria that survived.
When antibiotic-resistant bacteria develop in livestock, such bacteria can be passed on to those who consume the meat. And while this usually doesn't affect people due to the cooking techniques and other things, it can sometimes have serious consequences. Reactions can range from mild food poisonings-like symptoms to severe illness or death. E. coli and Campylobacter are concerns for the poultry industry in particular. Using antibiotics at such a high rate is unsustainable, as they will eventually lose their potency entirely. This would be a significant issue because humanity still relies heavily on the use of antibiotics. In addition, if this problem continues to expand, people will increasingly lose trust in the livestocking industry. This could have serious consequences for many people who rely on this industry for work and income.
Hello, my name is David, and I will explain the ways in which we can go about dealing with this issue. At this point, there is no permanent solution to this problem of bacteria becoming resistant to antibiotics in the livestock industry. However, there are ways in which we could better deal with this issue. For example, as it is, many of the livestockers have not done enough research into how much of a single antibiotic they should use per animal. In fact, 3/4 of all antibiotics in the United States are used for livestock, which means only 1/4 are actually used on humans. This is way too much. It seems that when more of an antibiotic is used, the bacteria seem to become resistant quicker. Therefore, diminishing and regulating the amount of an antibiotic that is allowed to be used would help to extend the life of that specific antibiotic, giving researchers and scientists more time to develop a new antibiotic that bacteria would not be resistant to. Also, once an antibiotic is no longer used, the bacteria begin to de-evolve and become no longer resistant to that specific antibiotic. For example, when ciprofloxacin was banned in 2005, it was found that 30% of C. coli were resistant to this antibiotic. But after being banned for just 5 years, the percentage of resistance had dropped all the way to 13.5%. This means that antibiotics could be recycled once bacteria was no longer resistant to them. Another way to help solve this problem would be to develop new ways of discovering if specific bacteria is resistant to an antibiotic quickly. If we can quickly identify when a bacteria becomes resistant to a specific antibiotic, then we can help to prevent this antibiotic that no longer works from being used. For instance, as soon as we discover that bacteria are becoming resistant to a specific antibiotic, we should push to apply a regulation on that specific antibiotic's use. In conclusion, this topic is directly related to chemistry because the antibiotics and how they are developed most certainly use chemistry. After all, these antibiotics and their development is no guessing game. This is also related to biology because the process that the bacteria go through to become resistant in the first place is something that cannot be expounded upon without the use of biology. Thank you for watching our presentation.